Thank you very much, everyone, for coming to our Innovative Discovery series. We have a treat today. It's my great pleasure to introduce Dr. Robert Atkins, our speaker. But before Atkins, I'm sorry. Before I introduce him, let me mention some housekeeping issues. For CME credit, as usual, you must sign in and you must include your email and credentials. Do not miss, please do not miss our March 1st Tech Talk with Mumita Bhattacharya from University of Delaware, and she's going to talk about work that she's done with our data here at Christiana Care, mostly related to patients with chronic kidney disease. So this is really, really exciting. Also, please note that the March 16th ID series will have a special guest, Dr. Yuan Yi from Purdue University. So please register to help us prepare. As usual, as always, the full schedule is posted on our website. And lastly, do not forget, the registration is still open for the Accel Community Research Exchange on March 12th. And the keynote speaker for the event is Dr. Georgia Dunstan, who is the founding director of the National Human Genome Center based out of Howard University. She's an accomplished speaker on health disparities and genomic research. So if you haven't yet, please, please, please register. So Dr. Robert Atkins, I'm really excited that he's talking today here. He has multiple titles. He's the director of the Center for Clinical Pediatric Research and Development at Nemours. He's also the head of Tissue Engineering and Regenerative Medicine Research and the chair of the Institutional Biosafety Committees for all Nemours campuses in the Delaware Valley and Florida. He's also the program director of the Professional Development Corps for our Delaware Accel CTR, and also the director of research for one of our other large program in Delaware, the Delaware INBRE. And he's also affiliated full professor at University of Delaware. This is quite a thing. He has a PhD in cell and molecular biology from University of Pennsylvania and has been continuously funded by NIH, American Heart Association, National Aeronautics and Space Administration, the Nemours Foundation, and many other foundations for many, many, many years. He has been a mentor for many trainees, and we all know his passion for mentoring and for also mentoring the mentors. And I must say that I've learned a lot from Rob. So he has a long list of manuscripts, book chapter, and also patents, 5 patents. So this is very impressive. So thank you again for being here. We are really honored, and thank you for giving the talk.
Thank you very much for the introduction. I welcome everyone. I appreciate seeing you guys here. I want to talk to you a little bit about some of the work we're doing up at Nemours, and I want to do that in the context of team building, how we're working across multiple institutions in the state of Delaware. So I have this long title, so I figured there's words in here for a lot of different people to come. So I'm going to go team building and translational science, chemistry, biomaterials, and improving recovery after vascular surgery. And toward the end, the study I'm going to talk about is a preclinical study developing materials to help, sort of regenerative materials, to help with precision therapeutics for patients that undergo congenital repairs or for adults even that undergo vascular surgery. I don't have any financial relationships to discuss. I hope that changes sometime in the future. I will talk about some work that's currently there's a patent that's being prosecuted, but there's no financial relationships yet. And I want to let people know that for those of you that have visited me up at Nemours before, our labs and offices have moved down to the E400 building at the DuPont Experimental Station. So we're really excited about that. We're getting things up and running, and I'm happy to give tours if anyone's interested in coming up there. And a couple of things that this move will do, we were fragmented. Sort of this group, the center that I run, was fragmented even in different buildings on the old Nemours campus, and we're all on the same floor or one floor above. So we have the 4th and 5th floors in the E400 building, which you can see here. So I'm going to talk about teams, and I'm going to start off by talking about why we bother with, because it used to be sort of when I was first starting off in science, it was kind of the lone wolf, go get your R01, be famous in your own right. But there's been a big shift over the past probably 10, 15, maybe even 20 years where success is really predicated by the people you work with and the teams. So this is from a study, and if I fail to mention where the data are from, everything I'm presenting is published by others or by us. So the references should be at the bottom. So this is a study that looked at relatively equivalent projects on tobacco utilization. So the blue, the diamonds, are teams. That's a cooperative grant, and the other two are different types or flavors of R01s. So what you can see, and what's pretty clear, is that early on there's a lag getting the team up and running, but then overall productivity, and this bears out in other disciplines and in other studies, productivity of teams is much higher. So these are all normalized, and we can talk about how this study was done, but what I'm trying to show you is that in the long run, the teams are far more productive. And in the context of clinical, so I work at Nemours, which is clinical pediatric enterprise. There's also a big benefit to the institution, and I always try to present this, to getting clinicians involved in team science. And this again is data from these, the top two, number one and number two at the bottom there. But there's an increase in the quality of clinical services at the institution. The flow of ideas between clinical and research fields, and an increased focus on critical clinical challenges. So the institutions that work on having research integrated with their clinical divisions see an increased focus on clinical challenges and solutions to them. There's better dissemination of evidence-based treatment. You tend to stay more ahead of the curve in other places. You can track funds and resources for capacity building. And there's a big one that's an issue that we're trying to really work on in the state, and that's retention of knowledgeable, talented, skilled clinicians in what's a competitive global economy for clinicians. So there's lots of reasons why we want to work on teams. Over the past, so these data are up to 2015. So since 1960, so I was born like here. So in my lifetime, we're just going to focus on the 4 to 10 authors publications. So on the legend here, it tells you how many authors are on papers and sort of the number of papers they're published with that group. There's some enormous articles, especially in the age of genetics, advanced genetics and genomics. But what you can see is that the kind of 4 to 10 author manuscript wasn't as common way back early on in my life as it is now. And sort of the size of the teams that are effective in getting publications out, that is sort of the sweet spot that people see. And that number is continuing to go up. So there's lots of reasons, better productivity. There's trends in the field. There's trends in funding. There's even trends that are being tracked in multi-PI grants, which are possibly better, at least as good in terms of the rate of their funding, but the dollars per PI is much higher in multi-PI grant, and the overall productivity is higher. So we like teams, so I'm sorry. So there's also been an increase from NIH's perspective. These are program grants and trends over sort of been years on the x-axis there. So the orange line at the top is the overall trends in funding. So the commonly awarded mechanisms, and this doesn't include P20. So our COBREs and the INBRI type mechanisms aren't included in this. CTR is a U54, so that's one here. So there's been a big increase from NIH, and the plans are to increase some of these other programs even more. So better productivity, better science, better engagement, especially if you include clinicians on your team, and access to funds. All good reasons to work as a team. Some of the characteristics of effective teams, and I've highlighted some of the things that we've seen as key and others as key challenges, are listed here. So effective teams have effective leadership and management. There's a good awareness of where you fit on the team and how everyone works together. Good teams are based on trust. In fact, if no trust means no team. You can have a group that's trying to work together, but trust is paramount importance. And one of the critical challenges in getting a team off the ground if you're just assembling one. effective teams have open communication. They're effective in building and making sure everyone shares expectations and knows what is going on, what the next step is, what their role is in the team. So clarifying expectations is also a critical challenge and something that we often overlook as we're sort of in the heat of battle doing research. There's shared vision, and that's revisited often. So as the group moves forward, it could be that some members of the team drop out. There's the vision changing a little bit, but everything is shared amongst the team. There's always provisions for recognition, especially for the junior faculty and the students and the fellows. And a big thing is authorship, making sure effective teams make sure that authorship criteria and everything's clear early on. And that is a stumbling block for a lot of teams. You get to the point where you're publishing and a key member of your team, two key members think they're both their article and they should be first, you've got a problem. And you should have addressed that much earlier. It's effective teams allow disagreement, but avoid conflict. And that goes back to trust. So if people trust you and they're willing to talk to you about problems they see, that's a great thing. And you really have to learn each other's languages, and I'll show you the team we're working with. So I have physicists, chemical engineers, one of whom very proud of the fact he hasn't taken biology since sophomore year in high school. And he's a great guy, high school. So we have to learn how to talk to each other, and that's often a big hurdle up front. And make sure we understand how to talk to each other at all levels, to each other, how their department works, so what the issues are with their department, how they're working there in their institution, and the regulatory bodies that oversee everything that we do. So it's an interesting thing working in a hospital, and I'm sure for the folks in the room here, sort of how little some basic scientists understand about IRBs and what you can and cannot do with patients and patient samples, and what HIPAA is, and sort of why you have to execute material transfer agreements and data use agreements. So there is a lot of communication and understanding each other around that, but effective teams do that. And they enjoy working with each other. And I really enjoy working with this. This is the team that I'm going to talk about their work. So you can find the surgeon. This is like, where's Waldo? That Ed is a PA surgical assistant that helps us with the he's an excellent research surgeon and has worked on animal models for quite a long time. Right next to him is Becca Scott, who is a fellow with an F-32. And her background is in material science and in immunology and biology. Millie Sullivan is in chemical engineering at University of Delaware. Wafa Amir is an optics physicist and biologist. Karen actually runs the lab, I think she might be on, and is a molecular biologist and a biologist by training. Prathamesh, who's right next to her, was in the chemical engineering department as a polymer person. Christy Kick is also primarily a chemist and a material scientist that I've been working with for a number of years. That's me. Thomas Epps, who's next to me, he's a chemical engineer also, who's proud of the biology training. And then we have a veterinary scientist and a veterinary technician who helps with the animals, a PICU fellow who worked on some of the stem cell stuff that I'll talk briefly about, another physicist and a graduate student who worked on ways to knock down on nanoparticles and ways to knock down some of the genes that we're interested in. So this is the group. The team ranges from hard scientists, physicists, all the way to biologists, which isn't really a soft scientist, geneticists, molecular biologists, chemists. And we need all of those folks together to sort of execute what we want to do. So what I want to talk about, the project we all work on has to do with the effects of stiffness in arteries and on cells that are associated with arterial function. So I'll give you a quick background, engineering background on what stiffness, what we're talking about. So if I mention modulus, or if I talk about kilopascals, Young's modulus and shear modulus are two things that I may conflate together, but they're actually two different measurements. So you can think of Young's modulus as the ratio between the force or the pressure that you have to put on a material. So think of a rubber band. So that's the force per area divided by the proportional change in the length of that material, right? So if you pull a little bit and it stretches a lot, that's a low modulus because the denominator is big. If you have to pull really hard and it doesn't move very much, the high modulus. Young's modulus, and those numbers are the same numbers as pressure, so we use pascals for that. When we actually measure some of the materials we use, we do it in shear. And this has come up as a communication issue between us and the engineers, because we're like modulus is modulus in the lab, in the biology lab. But there's a threefold difference. If you take a material, you measure shear modulus versus Young's modulus, there's a threefold difference. So if you ask a graduate student to make a material of a particular modulus, you should make clear which measurement strategy you're talking about. So it's a similar idea, except you kind of think of a column where you're pushing on the top, and how much does it distend? And it's the pressure, the force per area. In this case, it's in shear, and the strain, right, which is how much it moves relative to how much you pushed on it, which is given by this factor here, and you actually measure it based on the angle. That's the shear modulus. And just to give you an idea where these are Young moduli for a bunch of different physiologic systems. So blood and mucus, that's low, 50 pascals. That's the number we use. When you get up to kind of the tissues and the areas where we're interested in, and I'll orient you to where we're interested in a second, we're kind of in this region here. So from a few hundred pascals up to tens of thousands of pascals. And you can think of that as lung tissue, for those of you familiar with sort of the textures and how stiff tissues are, up to something that's like a chondrocyte or much stiffer material. Bone is really hard, that's something gigapascals. And when you get to something like glass or plastic, that's way off sort of the physiologic range, although we often use glass and plastic to grow our cells on. So the range we're interested in, sort of in here. And the tissue we're interested in is an artery. So we'd worked some in cardiac muscle years ago, but moved away from the heart a little bit, so not too far. So aorta and carotid, artery. And to remind you, arteries have a lumen that's lined with endothelial cells. That's the intima, which you can see here in this H&E. And then there's a sort of organized concentric layers of smooth muscle cells that can contract and sort of increase tone to tighten that vessel. And in some small vessels, they're frequently used to kind of constrict the size of the lumen. And then where we're going to focus is in this area called the adventitia. So that's the outer layer here of this artery. And the reason we're focused there, which I'll tell a little bit more about, is that the cells that live there are responsible in sort of a general way for the stiffness, the overall stiffness of the artery. They're also some of the primary cells that respond to changes in stiffness that happen either as we age. My arteries are stiffer. Younger folks in the audience who are stiffer than little babies. And that's just a fact of life. The arteries get stiffer. So some of the things that affect arterial stiffness in sort of a bulk way are aging. When you have hypertension, some metabolic diseases can stiffen your arteries. Connective tissue diseases, these are less common with Marfan syndrome. Poor cardiovascular tone and health. Sleep disturbances, which for those of you who are interested in sleep disturbances, there's a linkage between sleep and matrix metalloproteinase activities, alter stiffness in your arteries. And the one I want to focus on, which is the dot down here, this is from someone else's paper, is surgery. So when a surgeon goes in and manipulates an artery, whether they're doing it to get it out of the way, they're doing it to graft it, or they're just doing it because that's part of the procedure, there's a maladaptive cascade that's triggered in the artery that often ends up with stiffening, atherosclerosis even, and occlusion, depending on how much manipulation and how profound the maladaptive response is. So we're going to focus on that. That's the thing that we're trying to fix, right? So if with all these chemists and the materials folks, I want to come up with a way, and the stem cell folks and regenerative medicine biologists, we want to come up with a way that if a surgeon operates on a vessel, we can attenuate that maladaptive cascade and encourage healing. So you don't have to go in and redo the procedure or do a balloon angio. Like you don't have to go back in and do more. And to give you an idea of how bad the problem is, a standard mechanism for corn in adults, so data in kids, which is, I work at a pediatric hospital, data in kids isn't that great. We just don't know. There's problems. We don't have good numbers. In adults, getting a vein, a saphenous vein bypass graft, depending on where you are and the sort of the health of that individual, those grafts fail up to half the time. 50% of the time, the vein grafts fail. And a lot of that has to do with the manipulation of the vein during surgery and the altered hemodynamics. once it's in the flow, once the vein is connected. Arterial grafts fail at also high rates. I don't want to go down that road and talk about those data, but they fail at unacceptably high rates. And some, like a radial artery graft into your for coronary artery bypass grafting, they can fail 30 to 40% of the time. Right. So we think what we're working on is, one, understanding why the vessels stiffen and fail and get occluded. So I'm going to focus you guys on the left side of this figure, this diagram, where you can get an alteration in stiffness. And I'm emphasizing to think about stiffness in two different ways, and I'll tell you what those two different ways are in a second. But one way is elastin fragmentation. You lose the recoil. So the proteins that are responsible for the diastolic recoil in the artery, they get broken, right? So it's stiffening because it can't snap back. You can also stiffen because of fibrosis. And the primary, or one of the primary molecules that impacts fibrosis is collagen. All right, so here we go. Maybe not. Here we go. All right, so adventitium. So stiffening is a problem. Adventitium is involved in the physiologic process and maintaining the stiffness of an artery, but also in the pathophysiology of pulmonary hypertension, in sort of stress responses in arterial circuits. It's a pretty complicated tissue. So this diagram is from Kurt Stenmark's review. These layers, the endothelial layer and the smooth muscle layer, are more complicated than the cartoon shows, but he's an adventitium guy. So he's really focused on the adventitium. There's lots of different cells down there, including stem cells, adipocytes, fibroblasts, which you can think of as maintaining kind of the extracellular matrix and the stiffness, and being one way that they're described as immune sentinels. So if something starts to happen, immunologic or inflammatory response, the adventitial fibroblasts get activated, and then all hell breaks loose. So those fibroblasts produce matrix, right? Collagen in particular. They degrade matrix using matrix metalloproteinases, which can be inhibited by these inhibitors of matrix metalloproteinases. They make tons of different cytokines that are signaling each other, the other cells in the adventitium. Even the smooth muscle cells and the endothelial cells are driven by chemokines that are produced by adventitial fibroblasts. They communicate and, in fact, regulate, and even I'll show you some data on fibroblasts sending out signals to try and call monocytes into the tissue as part of a response. They interact with the vasa nervi and the vasa vasorum. Those are the nerves and microvessels of the vessel itself, and they interact with the endothelials and the epithelial cells. The adventitial fibroblasts, if you're looking for the definition, they're the primary cell types found in the vascular adventitium, and they're responsible for a number of physiologic and pathophysiologic roles. And we don't really understand them very well. And one of the things we don't understand about them is how they respond to these alterations in stiffness. So as you get older, your adventitial fibroblasts are living in a stiffer and stiffer material. And that's just sort of the general stiffness. There's also, if you think about blood going through an artery, there's this sort of rhythmic, every second change in a pulse wave that goes through the tissue that propagates differently. So the cells, as we get older, are in different stiffness materials. And these are some of the papers that we've had out recently looking at aortic adventitial fibroblasts. So I'm going to talk some about this one. It's from Becca Scott, who and actually all these folks were in that set of pictures. This is one of Becca's papers recently in Biomaterials, looking at how they respond to just sort of a static stiffnesses and changes in stiffness. And I'll talk about a study in an animal model. I have to tell you, there's going to be some surgical pictures coming up. I'm required to say that when I talk to college students. I'll say it to you too. You probably won't be as shocked as they are. But I'll talk about that. And that's Karen and Becca put that paper together. That's also from this year. And touch on a little bit some of the stuff that we're moving into using these nanocomplexes to attenuate maladaptive responses. These are all right now in preclinical testing. So we're not in humans yet, but we're looking to move towards humans fairly rapidly if we can. All right, so let's talk about the effects of substrate stiffness. So there's no cyclic 1 hertz, like every second change. We're not propagating anything through, but we're altering the stiffness of the substrate that these adventitial fibroblasts are in, and we use hydrogels for that. And I'll show you a diagram of what a hydrogel looks like, but it's basically a network, a polymer network that doesn't dissolve in water, but holds a ton of water. And I can give you example, some contact lenses. For those of you who have little kids, those little beads that fall out of diapers, they're hydrogels, and they can absorb a lot of water. And typically, and for the gels we work with, they're more than 90% aqueous, right? So the polymer doesn't dissolve, holds a lot of water, and they're very flexible, and you can get their sort of elasticity and stiffness to mimic natural tissues. And if you build them, so this is the polymer we use, or one of the polymer sets that we use. This is from Becca's, Scott et al. Down here, Becca's paper. It's a polyethylene glycol backbone, and we can mix these. This is a 4-arm star, so it's an X, polyethylene glycol and a thiol terminated. And then we can mix that together with molecules of interest that we want to put in for the cells to interact with, or to alter the stiffness, or to basically do anything you want with chemistry, which is pretty slick chemistry. And these molecules are all coming out of Christy lab. So these are malamide, that's this little structure up here, terminated. So this is an RGD sequence that the fibroblasts will bind to. This is an MMP2, matrix metalloproteinase 2 target sequence that the cells can interact with and cut. And they do that when they're moving around inside tissues. So if you mix together thiols and malamides, they click, chemical chemistry is click chemistry. They interact and they make a network. So these are our beautiful adventitial fibroblasts. You mix them in with these compounds, which have the viscosity of water. So you mix everything together, and a few seconds later they click, and you can get pretty much whatever chemistry, whatever binding sites, whatever molecules you want in there, and you can control the modulus, so the stiffness, right? So with a 5 for this system, 5% refers to the amount of polymer that's in the hydrogel. So basically, the other 95% is aqueous stuff and cells that's in the hydrogel. These are the stiffnesses in kilopascal. So by tuning the molecules, you can control the concentration of the binding sites or the cell interactive sites so that they're the same across these different weight percents, but drive different stiffnesses. You can put the cells into those, and you can ask the cells, well, what do you think at? Sort of in an anthropomorphic way. That's kind of what we do. So what we found, so I'm going to summarize data from this paper and then talk about another paper, and hopefully a couple of people might be able to suggest to me what's going on with our animal models and what's going on with these cells. Because we have some great data and some good ideas, but fundamentally we still don't understand how the cells are sort of thinking about and understanding these differences in stiffness. But in these gels, so 400 pascals, that's pretty soft, like really soft jello. And these are in shear, so multiply that by 3. You want to go back and you memorize that chart I showed you with tissue stiffnesses. So this is across the range of stiffnesses we're interested in and that the cells experience kind of on a regular basis. So if you put them in soft stuff, they proliferate more. And this monocyte chemoattractant protein is sort of the cells calling for monocytes and macrophage. So they initiate this sort of immune resistant inflammatory response. If you put them in something that's sort of stiffer and in the middle of that range, they increase their production of matrix metalloproteinase, which indicates they're going to start remodeling, right, and attacking that MMP target site that we put in there. And the number of fibroblasts that are becoming what are called, they're transdifferentiating to a myofibroblast. And we can, if anyone has questions about it, we can talk about the difference in function, but in like a crude way, for what we're trying to do, the myofibroblasts phenotype is bad. We want the fibroblasts to be sort of trying to heal the tissue, not trying to overtly make more and remodel the matrix too much. If you go a little bit stiffer, they tend to be more angiogenic and sort of calling for endothelial cells and for more sort of basovasoforms to start. So and we've known this for other cell types. Cells are exquisitely sensitive to the stiffness of the material that they're in. We just wanted to characterize it. So we wanted to know, well, how are they doing that? Like, what's the pathway? So that we can intervene with the pathway and better understand all the mechanisms that are associated with stiffness sensing in cells. And the primary way, and one of the major ways that cells responds to their environment, how stiff it is is with these integrins and receptor tyrosine kinase mediated signaling pathways. Right, so now we're getting into stuff that Millie Sullivan works on and that I work on and that Becca Scott's really good at and Karen knows about. But some of the other folks are like, not sure what you're talking about at that point. Sorry, Thomas, if you're listening. But basically, if you have a hydrogel, the cells can sense the mechanics of it, and they use integrin binding and what are called MAP. These are mitogen activated protein kinases, MAP kinase signaling pathways. And for the cells we're working with, these human adventitial fibroblasts, I should say those are, you can get them, they're from a 53-year-old man. There's 3 pathways that we were interrogating. So I'm going to refer to those as ERK, right? So this is one way that the cell might signal through and alter what the cell's doing and gene expression profiles. One is JUNK and one is P38. And we don't need to go into specifics, but those are 3 different ways that the cell can sort of manage the information coming from the outside into the nucleus, and there are specific inhibitors for each one. So initially the question was, which one? I was thinking, as were others, that the cells had kind of a rheostat. Stiffer, they turn this way, softer, they turn that way. So we did the study. Actually, Becca did the study. And what she found was it's not that simple. So these are now just the softest and the stiffest gels, kind of simplify the overall data interpretation here. And these are just 2 of the things she measured. So that MCP1, which is an important molecule in the tissue, because that's how the fibroblasts call monocytes and macrophage, and we want the macrophage that are eventually called in the patient. There's multiple flavors of macrophage. We want them to be pro-healing and not, sort of pro-inflammatory. So MCP1 is important. And when you look at the softer gels, the MCP1 amount, so the dark black here, that's dimethyl sulfoxide, that's the carrier for the inhibitor. So that's the baseline. And then you put the inhibitor on, and the cells respond to all 3 inhibitors and reduce the amount of MCP1 that they're using. But that right away is interesting. I thought they were going to have 1. But in the soft gels, they're using all 3 at different levels. When you go to the stiffer gels, kind of on the upper end of the range we're interested in, they do use only 1. And it's JUNK. They use the JUNK pathway to sort of understand the stiffness. But what that means is, in this similar kind of story for MMP2, they shift the pathway they use, understand that signal, how stiff the matrix is, to interpret it and express the phenotype. So what that says is, as is often the case in physiologic experiments, it's way more complicated than a rheostat, because they're not only changing their phenotype, what they're doing, they're changing the way they do it just by these sort of subtle changes in stiffness. So that's important. As far as I know, MAP kinases are not pharmacologic targets for blood vessels. They might be, but if this is a general, which I think it is, a general phenomenon, then you need to know the stiffness to be able to predict, well, which inhibitor do you want to use and which drug do you want to use? And some of the variability that we see in responses to drugs might be for blood vessels, might be associated with the physiologic condition that the target cells are in. So that's sort of a new idea, that it's not just the phenotype that's altered, it's the mechanism, it's the pathway that the cells are using to interpret. So this just summarizes some of the other things that Becca found and the group found looking at these human aortic adventitial fibroblasts. And it's a little tricky to figure out what exactly the cells are thinking. So for VEGF, there's somewhat of a shift. You can see that's column. VEGF is vascular endothelial growth factor. So that's one of the ways that cells stimulate endothelial cells and revascularization. I will tell you, in blood vessels, that's not necessarily the healthy way for revascularization. In fact, VEGF signaling by these cells is often associated with atherosclerosis. What you want the cells to do is use FGF2. Anyway, that's getting into the weeds, but they're pretty similar. So it doesn't seem to matter as much whether you're in the 5 weight percentile or 10 weight percentile at this point. Again, this is slightly different, and I'm jumping over a whole bunch of stuff. But for this data set, things like whether they're proliferating or not are very different, which pathways they're using, and whether they're calling with MCP1 or not, which we saw in the other graph. So we're following up on this, trying to figure out what exactly is going on and trying to tie it back to clinical and physiologic sort of observations in patients. But this paper just came out in Biomaterials last year. And then I want to switch and talk a little bit about the effect of substrate stiffness on vascular tissue. So this is the animal model. There are two, sorry, you're going to see pictures of surgery now. There are two generalized ways. Are there any vascular surgeons here? Okay, so there's two ways that people, there's more than two, which is why I asked that question. There's two general ways that surgeons prepare blood vessels for grafting. One is called skeletonization. And I didn't show you in that H&E early on, but they strip away all of the associated tissue down to the vessel itself. So this surface right here that you're looking at, that's the surface of the adventitium for this. This is an internal mammary artery that's going to be grafted, or they leave on what's called a pedicle. And this is a no-touch procedure you might have heard of. And surgeons pick which way they want to do it. There's some art to it. There's some science to it. There are clear advantages to skeletonization, especially for thoracic surgery where you're taking an IMA, internal mammary artery, for bypass grafting. You get a longer graft. It's easier to visualize if there's any problems. There's decreased actually post-surgical complications, especially infection. But there's a big disadvantage that there's an increased chance of sort of maladaptive response and atherosclerosis. So what we're trying to figure out is, well, is there a way you can get the advantages of skeletonization, keep the advantages of skeletonization and use these materials, right? If you can design them well, they have the right stiffness, the right modulus, include stem cells and anti-inflammatories so that you can put a synthetic pedicle back on. So that way you get the benefits of both. You get the benefits of skeletonization without the increase in failure and atherosclerotic risk. So the idea is that for the first study that I'm going to talk about, the long-term idea is we'll use a rabbit model with a carotid to jugular transposition interposition graft. And we'll have a whole set of hydrogels that we'll try based on our assessment of the stiffness of tissue going in and sort of the patient's condition when we do the surgery. But the study I'm going to talk to you about now, it's just hydrogel. It's pretty simple hydrogel. So the artery is skeletonized and we either leave it or we inject a hydrogel around it. This shows you down into the right. Think of sort of like an epoxy, a two-barreled syringe that has two low viscosity solutions. It goes through a mixing tip, and within a few seconds, it clicks. That chemistry is very fast, and it goes to the modulus that you want, and then you close. So from a surgeon's perspective, it takes an additional about 30 seconds on the procedure. So that's actually something you can expect to do in an OR, and we're practicing preclinically doing that. So this is, we operate on the left side. This is rabbit anatomy. Because, and just so you know, the animals are fine. Because of the circulatory physiology and what's called an intact circle of Willis in the rabbits, you can take down the whole left side. So you can just clip the common carotid artery on one side. The head's completely perfused by blood coming up from the other side. So for that reason, it's a good animal. The size of the arteries is similar to what surgeons use in humans, and sort of some of the responses, physiologic responses in the tissue are very similar in rabbits. So we always operate on the left side. And this is, I took the video out because it was too big, but we use high frequency ultrasound. So these are, for those of you who do ultrasound, these are driven at 40 to 45 megahertz, and we can go up to 80. So you can get a voxel resolution with our animal ultrasound around 30 microns. So right now we're just using it to measure what's called a circumferential strain. So if you look, this is just a picture, and we cut a line right through, that's the carotid artery right there. And this is a time sweep. So when the heart beats, the bolus of blood goes in. So this is over time from left to right. So the artery gets bigger, and then there's this diastolic or elastic recoil, and blood gets pushed through. And you can measure these dimensions systole and diastole and calculate what's called a strain or cyclic strain. We use the green Lagrange vector. I don't think there's any engineers in this, well, maybe on the phone. So this is the formula that we use, and it's expressed as a %. So one of the surprising things we found right off the bat in doing these initial experiments, well, it wasn't surprising that the strain was at 10%. That's about what it is in a carotid. These carotids are small compared, yours is bigger. This is a 3.5 kilo mammal rabbit. So it's, a couple millimeters across. That's what you expect. And that strain stays, and we compare contralateral side to operated side. But within 72 hours of skeletonization, and this was the surprising thing, we get this is physiologically a huge depression in elasticity. So the compliance or the circumferential strain drops almost immediately. So all our plans to do high-tech stem cells and anti-inflammatories and drug delivery got set aside so that we can start figuring out what's going on with this first step. There's not a lot of studies looking at skeletonized arteries, because usually when you skeletonize an artery, you're going to graft it. So it's difficult to discern what's an effect of the graft and altered hemodynamics coming after the graft and what's an effect of skeletonization. But at least in the rabbit carotid, we get this consistent and persistent. We've gone out, I think, 8 weeks now, and it doesn't come back. It drops way down, 6%, and it just stays there. Right? So the treatment we were going to do, the intervention, was to use a simple hydrogel. So this is just style and malamate terminated, no cells or anything mixed in. It clicks together. Doesn't actually, it erodes away inside the animal, but it doesn't degrade. We don't put anything in there for enzymes to degrade it or anything. It's pretty simple, but as simple a hydrogel as you can think of. And we picked a weight percent. So this is a 4 weight percent hydrogel, which has a shear modulus of 4.8 kilopascals. So during surgery, after the skeletonization, it's injected. The two solutions are on two sides of the syringe, and it gets injected around the outside. And I should mention that this is in a paper that Karen Robinson and Becca Scott are co-first authors on that came out last year. Right, so we inject the hydrogel around, and surprisingly, or I should say, as expected, that simple hydrogel protects the circumferential strain in the vessel. So we started seeing this, I think, the end of 2016, and spent most of last year, first half of last year, trying to figure out how that's possible, what's going on in this particular context. But when you look at so typically when an artery starts to fail or gets occluded or stiffens, there's an increase in the thickness. I mentioned the intima is this endothelial layer on the inside, and then there's a medial layer that's smooth muscle cells, so that gets bigger. And this is, so up top is a H&E stain, and these little yellow lines, and if you can see them, that's where we measured, or Aaron actually measured across, so you can calculate that intimal medial thickness. And down below is a stain for smooth muscle cells. We're sure we're measuring the right thing. And skeletonization does not affect that, not within 21 days. You have to go much further out for those kinds of bigger cellular responses to kick in. And this is as expected. So we wanted to look more deeply at the tissue. So we turned to our physicist friends to do 2-photon microscopy. So we want to be able to look at the elastin, right? The elastin is the stuff that makes it snap back, and the collagen, that's what stiffens the vessel when it increases in size. So there's a way you can do that without staining the tissue, and then you can use immunohistochemistry or other histochemistry afterwards. And I'm not going to go into great detail, but that uses 2-photon microscopy. For the elastin, you kind of put 2 photons, they focus in, they actually combine, so the wavelength changes. The elastin absorbs some of that and then releases a different photon. So it's fluorescence, it's autofluorescence. But there's a decrease. There's not a direct relationship, or there is a direct relationship, but there's a dissipation in heat. So the energy coming back out is lower than the energy you put in, which for light means it's a different color, right? So we put in the light and it comes back out, and I'll show you in a second, it looks yellow. It doesn't look yellow necessarily on the scope. That's the color. We're going to pseudo color it. In second harmonic generated signals, so this is an energy conserving way that you can do 2-photon microscopy and look for molecules and structures that have particular interactions with the light. Collagen is one of them, but the light's actually conserved. There's no light, there's no loss, there's no heat, there's no anything. It just pops back out at half the wavelength. So the color again is different, and the color between this emission are different. So you can look at, this is a carotid artery now stain. So the yellow that's showing up here, that's elastin, and this is a control, pretty typical rabbit carotid artery. you get this internal lamina right underneath the endothelial cells, and then layers of elastin, which is exactly what you expect. We can do the second harmonic generated signal in a way where we look at forward and back scattered light, and then use that to figure out something that's, you figure out what's going on with the collagen. All right. So when I had to brighten this for you guys to see in the room. So this is the control on the left. This is a skeletonized vessel in the middle. These are at 21 days, and this is a hydrogel protected vessel on the right. And what you notice about the skeletonized vessel is that you start losing those beautiful laminae. And in fact, in that image I showed you, that cartoon about things that make your arteries stiffer, elastin fragmentation is one of the main. So again, for some reason that we're still trying to get the specific molecular kind of biological reason for, you get fragmentation of the elastin with skeletonization of a carotid artery. If you put our hydrogel on, you don't. And that's quantified down here. We use you might be able to see that there's cyan and magenta signal. I might have washed it out when I brightened it. But those two colors allow you to calculate differences in collagen. So there seems to be a difference in collagen turnover. It's not as big an effect, but probably both things are happening within 21 days where the recoil is down, the elastin's gone, and also the artery's getting stiffer at the same time. So we're moving ahead with experiments to try to understand that. It's not the only thing going on, and this is my last slide coming up, and the tissue. So we can protect the elastin and sort of keep some of the biomechanics of the vessel with this simple hydrogel application. But remember I said the fibroblasts get activated when surgery or something happens. They're still activated. So they're probably activated as soon as the surgeon cuts tissue, they get turned on. So we're working on ways to not only, with the hydrogel, we can protect the biomechanics, but also deliver molecules to knock down pathways that are triggering maladaptive responses. So this is from Chad Greco, who's now out in California, and Thomas Epps and Millie Sullivan in our group, so our team working together. And I'm not going to go through in detail, but what Chad did and showed was using these high-tech polymer and chemically specific nanoparticles on cells that are cultured in a dish. We're just starting to do this in animals now. He can use light to knock down gene expression, right? So this trace, just think of these traces here as being the amount of expression of this particular target gene. When you hit it with light, it drops down. And then after, these are hours, so after a certain amount of time, that dosing is done and the gene comes back on, you can redose by hitting it with more light. So right now the light is close to UV, and Thomas is working on ways that we can use longer wavelength light, and in fact may be able to do this transcutaneously, so that you can introduce a material that allows you to biomechanically stabilize the tissue and then knock down these inflammatory cascades early on in the progression toward failure of the tissue, and then you kind of walk away, as we think everything bad that happens in these vessels happens early on. You kind of push the first domino over during surgery or immediately after surgery, and then a year later is when the vessel fails. If we can stop that initial cascade, we may be able to keep these patients from needing follow-up. So what we're going with this is to look for an improved understanding of fundamental mechanisms relating stiffness to cell and tissue function. We're preclinical testing of these simple hydrogels and arterial grafts, and we're starting to preclinically test hydrogels and more complex hydrogels on vein grafts as well, so not just skeletonization. And we're working on industrial partnerships. I mentioned there's a patent filed, and to begin, which is something that would be brand new to me, getting an IND and IDE, for those of you who are familiar with those processes are. My initial conversations with FDA indicate they're going to look at this as a drug and a device, so that's fun. So we get to go through both of those pathways. But we're moving in that direction because the simple iteration that we showed the protection for, and here's the team again, we showed protection for that molecule. Polyethylene glycol has been approved in lots of contexts. There's lots of pegylated compounds and PEG-based materials that are put in humans now. So at least that part of it, we're fairly sure the FDA is not going to give us a big problem about. We still have to do all of our second animal testing and get everything ready to help. But we're hopeful that we can get a simple hydrogel into clinical testing pretty quickly. So I do want to thank funding. We funding from Nemours, For this particular project, a good chunk of the funding comes from the National Heart, Lung, and Blood Institute. We've also had funding from the Swank Foundation, Center for Advanced Technologies, American Heart, and some other places for other projects. I want to thank all these guys. This whole team is a wonderful team to work with. Not all teams are wonderful, but these guys hit on all those things for an effective team. And these guys are great. And I want to thank you guys for your attention, and I'd be happy to answer any questions. All right, who's going to tell me how it works? Yes,
so I'm on a team of sorts, I guess, with you, but I had no idea really until today what you do outside of mentoring. So this is really interesting. And I think I was able to follow along fairly well for behavioral science. My question for you is more process. So we actually, some of us heard a talk from our vice chair of medicine yesterday about how he formed teams and tackled problems within the health system. And the nature of the problems he was working on were more process improvement, much more right at the bedside, clinical kinds of things. And you're much more obviously in the realm of the translational work. And so for those reasons, it's obvious why his work is going to be in the operational space, and yours is, here's a problem that our surgeons agree is a priority. We're going to take it over here and work on it for a while. So I understand that distinction, but that put aside, can you speak to, because I think we face similar challenges, those of us engaged in research here as you are at Nemours, in a primarily clinical enterprise, some of the strategies you employ to kind of get that, I also understand your funding, but to kind of get that buy-in to be able to form a team like this and go work on things for a while until you can get a good answer.
It's a great question. It's challenging because, especially for I'm a trained researcher. So when you're trained as a researcher, you find a question that you want to answer and you go get funding to work on it. But from a team perspective, especially teams with clinicians, you have to make sure it's a win for everybody and everyone's excited about the project. So there was tweaking around what we were really going to do with this project. So and to give you sort of I'm not trying to skirt your process issue. I'm going to talk a little about the history of where these regenerative therapies sort of came from. This all started with myocardium, which is a hot area for post-infarct recovery and injecting stem cells, and you have to mix it with hydrogels. And the pediatric folks are not interested. Kids have heart attacks, but that's not the main thing. Main thing are congenital malformations. They need better recovery from surgical manipulations that they do. They have to go back in and do revisions. There's fibroses all over the place, and it's a nightmare. So I learned, talking to them, well, I'm at the hospital. I want to be at the hospital. We have to switch and work on something that they're interested in. And I think that's the key. It's not, it's a process step where you're meeting them halfway and finding something. It is as challenging as working in myocardium, but this study has implications for congenital repair also. So it was, and then pulling the rest of the team together. Christy is one of the first ones I found, and she's crazy enough to listen to me, and then maybe we can make these gels and do things, in animals and then eventually in patients. But you find engineers who are jazzed about the biological applications of what they're working on, right? So some of the things from the clinical perspective, and I didn't really talk about adhesions. But one of the ideas with this hydrogel around a surgical site is it'll block adhesions. And we sort of see that it's still a mess when you go in to dissect, but you can limit adhesions with this. So we're able to sort of get this material that accomplishes multiple things along the way. But we also need to knock down, there's this entire cascade of induced, these cells get activated because you're touching the tissue. They just turn on, and the gel's not. It seems to help us a little bit with that, but it's not going to attenuate. You turn them on, they're calling for monocytes and macrophage, and there's neutrophils. There's this inflammatory cascade. So the other thing the clinicians want is a way to dampen that inflammatory response. So we have a grant pending where we're going to try that with sirolimus, and there's some drugs out there. But the future way to do that is with stem cells and with these high-tech drug delivery mechanisms where you do everything in one procedure. You go in. For me, if I'm getting a bypass graft, they draw some blood, they get the stem cells, they tune up the material they need, they mix stem cells back in, throw in some polyplexes that we've identified based on like a precision medicine profiling, takes a certain 30 seconds more, and boom, you're done. So it was that process of sort of understanding what is it that the surgeons really want and what is it that they're looking for. So when I go and talk to them, they're like, when can we use it? And that's great. And then from the engineering side, Millie and Thomas are really interested in this light activation of polyplexes and how can you knock down in really like temporal, spatial controlled way, knock down gene expression. So rather than knocking down gene expression in the whole person and the whole organism, or in the whole neck or in the whole chest, you can precisely do it. You want it at the anastomotic sites because that's where the big problem is. Okay, well we can do that. We can put this material everywhere, but we only activate it with light at the site of the anastomosis. So it's finding where that ground is and being willing to say, well, my main thing is cardiomyocytes, which is what I would have said years ago. I don't think we've done a cardiomyocyte experiment in years, and they're still on the back burner. But we got a lot of traction out of this, and we're able to get a lot of partnership, and then the team is able to move together ahead. And when you get that feedback from the rest of the team, that helps the whole thing sort of gel together. It's tough. It really gels. It's tough to do. It really is tough.
So, a different question. One of the issues in patients with chronic kidney disease, for example, is arterial fistula.
Yeah.
So that they can be hemodialyzed efficiently. And the way to do it is to link the arteries to the vein so that the vein, in fact, have a better understanding of the pressure. And you know that. Is it a domain where, I think there are a lot of applications that we're working on that could be great for, and it's always a major issue for these patients because they have stenosis, they have at the link of the fistula, where the fistula is made. Very often they have stenosis, and then they have all kinds of complications. And it's really a question of life and death, because if their fistula doesn't work, then it's really hard to dialyze. So and I see a lot of applications from what you are doing.
Yeah, so that is one of, we have, with a team in particular, you have lots of ideas. So there's one of the applications that we really talked about. And in terms of the number of patients and sort of the need for some approach that allows that fistula or allows that access to be stabilized, if we can figure it out, right? And we're still sort of at the beginning of figuring it out. The procedure would be, okay, you establish the fistula, you put this high-tech material around the outside, and you have less fibrosis and sort of less stenosis. I'm not sure we could stop it completely because of the number of times you're kind of accessing, and especially for the dialysis patients, it's similar cell physiology. The tissues respond, the arteries, the veins respond a bit differently, but the arterial side responds with this sort of maladaptive cascade that gets you fibrosis. And the stenosis often comes, and I haven't looked into the literature on the fistulas, but I assume this is also true. The stenosis comes a lot from the adventitial fibroblasts responding to this insult, right, this stress, and inappropriately telling the other cells you have to proliferate. There's probably some physiologic growth context where that happens and that needs to happen. So if we can go in and stabilize the fistula and essentially tell the fibroblasts, well, knock that off. Don't tell them to do that you should be able to reduce stenosis. So that's sort of, it's right now kind of pie in the sky, and we're in animal models, but we have to get more grants. So we have more fellows, clinical fellows, and sort of folks that want to try different things. And even the cardiology fellows, they want to try different assessments so we can look at pulse wave and more Doppler imaging and get away from what is fairly simple ultrasound. But we have to be able to get the funding to do it. And I think once we're able to establish the funding, I'd like to come over and talk to the clinicians. I don't know what the pediatricians think of that project. They're all congenital heart repair folks that we've been talking to, but there's a huge need for it.
Okay, even in the kids, they are. Yeah. But here we have vascular surgeons and big population who really are taking care of these patients. So you want to go to the adults.
Yeah. I don't look at it as adult size. They might tune the materials differently, and maybe that's just the way I think. There's a trajectory. So the cells know how old you are because of how stiff the matrix is, and sort of how long their telomere lengths and how long they've been around. But as far as we can tell, and there's some people looking at these cascades, the cascades are similar. It's just, bless you, it's just, what do you have to do to get a 75-year-old cells to respond might be different than what you have to do for a 5-year-old. But if it's all within tuning to modulus, what kinds of stem cells and what dose, if it's sirolimus or whatever the drug, then you just dial it up. You dial it up in the OR or in the procedure room, mix everything together and inject it.
That's fascinating.
Thank you. Yes, sir.
One thing we've observed, whether it's trying to knock something out with an expression, trying to knock down the expression, or with targeted therapy, is we tend to look at that target and measure the effect of that target without looking at what the impact is on the rest of the system and how it's actually adapting to the fact that that target's being modified. I was wondering if you're looking at how the expression patterns are changing for anything else that might be impacting secondarily that pathway, or also lead to other longer term effects that you're not going to see with that short term impact.
So we have some pilot data that I'm not comfortable with yet. So we're starting with just expression arrays and looking at the RNA level. And we're especially interested in what the effects of skeletonization are, and if there are identifiable gene expression, RNA differences, what are those are affected by skeletonization and which are not. So there are some interesting hits that come out of that list. One of those is interleukin beta 2, and one of those is MMP2. So, but it is still early for us to figure out, well, what are the targets we want to measure. But part of what we want to do in the context of the hydrogels themselves, but more specifically in the knockdown. So if we were going to pick, and one of the targets we picked was interleukin 1 beta, because of its role in sort of the top of these cascades, and because we saw it in expression differences. But we really have to be able to interrogate within the anatomic region that we knocked down. Did we get it knocked down? And then were the targets downstream also impacted? And then whether there are side effects or there are sort of ripple effects, what else is going on and how long do those things last? So what Chad down on the right, what he was able to show is that you can do this in a relatively narrow window, so 60 hours, and then expression comes back on. So the effect of the polyplex is clear, so at least in our initial testing. So we think we have all the control, like all the ability to control it. We just haven't started measuring yet. But it's going to be, as you know, it's going to be complicated, and it's going to take. We don't even know what windows to look in. So we take snapshots where we can, even with the ultrasound. We take snapshots when we can. And I would, so when we get to that point, I'd like to ask, find out from you, where should we be looking? Like, do you do every hour for a little while, and then twice a day, and then once a day? Like how do you figure out where to measure? Because if you measure at the wrong time point or at the wrong frequency, you might miss the thing that's the most important thing for you to measure. But we are starting to head down that road. I didn't put any of those data in here because they're really preliminary. Yeah.
Just a question about your team. So when you describe your team, you've successfully been able to build a team across institutions versus just sort of within your institution. Can you give us the 30 seconds of what were the major barriers when developing work across these institutions, and then how you overcame?
So the main institutions in this cluster here are University of Delaware, Delaware State University, and Nemours. We're hoping to pull the adult Christiana people in. So some of the main problems had to do with, we want to be able to evaluate human tissue and surgical plans. But we need to do that evaluation at, say, DSU, which has this big optics facility, and the physicists do all this high-tech analysis. It's pretty challenging just to get the data transfer agreements and sort of all of that kind of paperwork stuff done so that you can work across the institution. And then the other part that's tricky is when you're across institutions and you're across disciplines, and sort of the way, this is Thomas Planchon, who's a physicist, the way he thinks and the way he kind of works on problems and the way he was directing his students was great, except it wasn't answering, initially wasn't answering the question that we wanted to answer. We had to kind of recircle back and go back and sort of think more about what are the physiologic, what are the things we want to know that are beyond the signal and the physics of the signal and sort of the ability to analyze the signal. So those are the two main things that were hurdles. There was communication across disciplines and sort of, and I can give you another example, and he's not on here because it was a different study between the hospital and the engineering college, but it's just it's understanding each other, the same language, same expectations about what, you know, when you say, okay, I'm going to take care of that experiment, and I'm thinking the experiment is X, and you're thinking the experiment is Y, and we get back together, and I'm like, what are you talking about? And also the interinstitutional, we can't send you that tissue for 3 months because we have to go through this whole process, and we have to consent people, and we have to get it approved, and the other institution not really understanding why they can't just get it and look at it. When Christy and I, one of the things we were working on, and I didn't talk about it here, was drug delivery using these hydrogels. So there's challenges in adults and kids in particular. Like adults, about 30% of adults don't take their freaking meds no matter what. Doesn't matter if it's injectable or they're just not adherent, especially if it's chronic. So for some targeted applications, oh, she's right there. So some targeted applications, we think the way around that is don't make them inject themselves twice a day. As they're leaving, the physician they're checking out with does a subcutaneous injection of this high-tech polymer. So we were going after thromboprophylaxis, and so it's a cardiothoracic surgeon is no longer, this is years ago, more is in me in Christy, because we're in this animal study. So that's the team, 3 people. And I think the first meeting, I quickly realized my job is to listen to what Christy's saying from the engineering perspective and the polymer design perspective. Look at Chris, who's the surgeon, and say, what Christy said, and translate it. So that is a challenge across disciplines and across institutions. It takes work and focus, and it takes time. And I think on that slide I showed about productivity in teams, that lag is because you got to get all that stuff working and make sure that everybody understands each other. But I would say those are the 2 biggest things across institutions. And if you're going to do that, don't let it lie for very long, thinking that things are moving ahead. You have to keep on top of the agreements and the consents and how many patients we have and all that stuff going on. Right. Thank you all very much.